All right, in this section, we're going to discuss methods of separating mixtures. We've talked in previous slides about how mixtures can be separated by physical means, and a couple that we highlighted way back at the beginning was distillation and evaporation. But there are others, and we're going to talk about them in this section. Mixtures can be separated by a number of physical means. We'll discuss in this section chromatography, which separates molecules in a mixture based on their differential distribution over a stationary phase and a mobile phase. A lot of information there, but you'll see what we mean. Distillation, which separates liquids based on differences in boiling points. Evaporation, which separates a soluble solid from a liquid. Filtration, which separates an insoluble solid from a liquid. And centrifugation, which separates mixtures based on differences in densities. There are times when mixtures need to be separated by chemical means, and there are ways to do that. The first is called gravimetric analysis, which is based on first removing a specific ion from solution by forming a precipitate of it. And the second is by chemical reaction, which removes a substance from a mixture of solids by causing one substance to go into solution by chemical means.
Filtration separates a solid from the liquid in a heterogeneous mixture. Filtration is the method of choice to remove solid impurities or components from a liquid. It can be performed by applying a vacuum as well to speed up the filtration. Filtration involves pouring a mixture through a specially folded filtration paper supported on a funnel and letting gravity pull the liquid through the small pores in the paper, just like a coffee filter. Chromatography separates components of a solution based on their unique physical characteristics. Molecules vary in polarity, size, and binding affinity. Any of these characteristics can be used to separate molecules into different fractions using different chromatographic techniques. The best technique for each separation problem depends on the types of molecules in the solution. Chromatography may be used to identify or quantify the molecules in a solution. It can also be used to purify the components of a solution. Two types of chromatography will be discussed here. Chromatography is the technique of separating chemical mixtures into individual components. The mixture is first dissolved in a fluid called the mobile phase. The mobile phase is then allowed to pass through a stationary phase. The various parts of the mixture travel at different speeds through the stationary phase, causing them to separate. In paper chromatography, the stationary phase is a strip of paper, shown here and also on this picture over here. Capillary action draws the mobile phase up through the paper with different components of the fluid traveling at different rates, which means they're separating. Each component is revealed by its color appearing at a different height.
All right, so let's go through the steps of paper chromatography together. So in this apparatus here, a drop of the mixture is placed 1 centimeter from the edge of the paper. So if this is our paper, we might have a drop down here near the edge of the paper. The mobile phase, which is a solvent, okay, some suitable solvent, is poured into the container, in this case to a height of about 0.5 millimeters. So you just want the tip of the paper touching the solvent. The paper is placed in the solvent with the sample spot above the solvent. So you're not submerging your drop into the solvent. You're just putting a tip of the paper in there. Capillary action, which you've all seen when you see water spread on a paper towel, if it's in like a little drip of water, that drawing of the water in is that capillary action. So capillary action draws the mobile phase up through the paper, the stationary phase. They travel at different rates, separating along the paper. So you can see whatever was in that drop begins separating into these different colors on the paper as the solvent moves up the paper. Each component is revealed by its color appearing at a different height.
Column chromatography is a general term. It refers to using a glass or metal cylinder to hold a stationary phase in place. That's called the column, while a complex liquid or gas sample moves through it and gets separated into its components. The column matrix is chosen to separate molecules on the basis of size, polarity, or affinity for the matrix, whichever quality is best for the sample mixture. As a sample moves through the column, and here you have a sample, and we're representing the different molecules by their colors there and sizes. Different components of the sample are slowed down to different degrees depending on their physical properties. So they're moving down like this, and hopefully you can see the red is at the bottom, green here, and blue is moving the slowest. Individual fractions are removed from the end of the column, down here, drop by drop, and contain molecules that are separated from the mixture based on their characteristics. Distillation is the process of separating a liquid mixture into its component parts. It can separate liquids from non-volatile solids or separate the liquids that comprise a mixture. The liquid is slowly heated until it reaches the boiling point of one of its components. As that component boils, its vapor is collected and condensed back into a liquid. But now that liquid is comprised of just that single component of the original mixture. The process is continued until each component is collected, separating the original mixture into pure samples. So here we have our original fluid, and that's in the distilling flask. It's heated with the Bunsen burner. You have a liquid evaporating like this, and then it's cooled in the condenser. You have cool water in here, it flows through, comes out this way. Now this water is not connected to the gas that is condensing here. They're separate. And then finally, the gas turns back into a liquid, and it's collected over here, and it's called the distillate. The receiving flask can be connected to a vacuum to support a lower boiling point to avoid decomposition of the mixtures if necessary. Gravimetric analysis determines the mass of a solid substance produced, usually by mixing two liquids or aqueous solutions. Precipitation gravimetry uses a precipitation reaction to separate one of the products. The solid product will be filtered using quantitative filter paper, washed and then dried. The mass of the solid recovered will be used to calculate the percent yield using stoichiometry of the reaction. The recovered solid product, which is up here in the filter paper, can be ignited along with the filter paper to convert to a stable oxide or sulfate to make a quantitative analysis. And it'll fall down here. You can see we have a vacuum here helping it fall. The special filter paper will burn off without any residues.
All right, let's discuss how we might separate solids by chemical reaction. When a mixture of solids contains one substance that will react with an acid or base, that component can be removed from the mixture by a chemical reaction. For instance, we've talked about brass before. It's an alloy that contains copper and zinc. Copper reacts with nitric acid while zinc does not. So the copper can be removed from the brass by reacting it with the acid. So here's brass, here's the acid. We put the brass sample into the acid and we see this reaction happening. The blue color that appears here after the reaction indicates the presence of the copper ions. So the copper is left behind here and is no longer in this sample. This is no longer a sample of brass, right, because the copper has been removed from the mixture.
Decantation. Decantation can be used to separate a liquid and solid mixture or a mixture of two immiscible liquids. What is immiscible? Well, that's liquids that will not blend together, like oil and water, that have different densities. For liquid and solid mixtures, the liquid can be poured off the top. For liquid and liquid mixtures, it is best to allow the more dense liquid to drain out of the bottom using a separatory funnel.
Here are a few examples of decantation, beginning with biodiesel. The production of biodiesel requires the decantation process to remove glycerol, which is formed as a byproduct during the production. Since glycerol is more dense than biodiesel, it can be separated through decantation. Crude oil processing also uses decantation to remove impurities such as mercury. Mercury is a very dense liquid that can be found in high levels in crude oil. It poses several problems during crude oil refinement by generating toxic waste, damaging equipment, destroying catalysts, and/or presenting serious health impacts. And so it is important that we remove those impurities as a part of the refinement process. Cream, like the cream you would put in coffee, is also obtained through the process of decantation. After centrifugation, the less dense cream floats on top of the skimmed milk.
Centrifugations. Another way to separate mixtures is with a centrifuge, and that's over here. A centrifuge is usually driven by an electric motor moving in a rotational motion. Samples, hopefully you can see them here, are spun until a centrifugal force pushes the more dense substance to the bottom of a tube. Centrifugation can be used to separate skim milk from whole, blood cells from plasma, uranium-238 from uranium-235, and even to separate your clothes from water. Pelleting happens when particles are concentrated at the bottom of the tube. The liquid above the pellet, called the supernatant, is often used in industry for a variety of purposes. Crystallization is a process in which a chemical is converted from a liquid solution to a solid with highly organized particles. There are three different ways to accomplish this: evaporation, cooling, and precipitation. Filtration or decantation might be necessary to complete the separation.
All right, let's discuss the steps of crystallization by cooling. So first, we have an impure solid here, dissolved into this solvent. We just stir it up to dissolve it. Next, that solution is heated. So we put it over the evaporating dish over a Bunsen burner to heat the solution and evaporate as much of the solvent as possible. So we're just driving off some solvent particles here to speed up the process. As the hot solution cools, we remove it from the Bunsen burner, we see crystals begin to form, and we can pour off this cold solution and be left with these crystals. If we pour it, through a filter, then the crystals would be left behind on the filter paper.
Crystallization by means of evaporation begins with a supersaturated solution. This solution is allowed to evaporate, leaving behind crystals. Supersaturated saline solutions are often evaporated to mine crystallized salt. In some places, like the Bonneville Salt Flats in Utah, vast expanses of crystallized salt are formed by evaporation. In some cases, crystals are precipitated by seeding a supersaturated solution. Making rock candy is an example of crystallization using precipitation. During this process, a string or a wooden stick, there's the wooden stick there, is placed into a supersaturated sugar solution. Sugar molecules stick to the string or the wooden stick, and these initial molecules act as a seed for larger crystals to grow upon.